Hello and welcome everybody. My name is Ali Beatty and I will be the moderator for this webinar. I would like to introduce today's topic, which is optimizing bioprocessing with Clickmers, improving flexibility and efficiency in purification. The presenters today will be Elisa Merklinger and Stephanie Bush. Elisa is a biochemist and works at Clickmer Systems as a senior scientist. She is responsible for assay development and other R&D projects. Stephanie has been with Clickmer Systems for over 6 years as a senior technical assistant. She is involved in the selection and characterization of Clickmers and contributes to developing their potential applications. Before we begin, just a little bit about APIS assays. APIS has established itself as a leading biotech company in the northwest of England, with headquarters in the heart of Manchester and business entities in Bonn, Germany, and Belgrade, Serbia. APIS has established comprehensive diagnostic development capability, acquired a bioinformatics and software capability, developed specific biomarkers capable of transforming disease management, expertly delivered on contract development for clients, and established its reputation as an international developer of transformative diagnostic solutions. With that said, today's topic is all about what innovations in bioprocessing are needed for improving yield, efficiency, scalability, and cost effectiveness. During the webinar, we will introduce Clickmers, which are versatile synthetic affinity reagents based on chemically modified single-stranded DNA. Hopefully, by the end of today, you will learn more about the fundamentals of Clickmers and their potential in bioprocessing and how they can be specifically developed for the needs of your production process. Remember to send questions in the chat throughout the presentation and we'll make sure to answer them at the end during the Q&A. And without further ado, I'd like to introduce our first presenter, Elisa.
Thanks for the intro, Ali. I'll pick it up from here. You might encounter challenges in your bioprocessing approaches. And what we would like to demonstrate today is how Clickmers can, for example, lower the variation between batches of material, meaning more consistent manufacturing processes, how they can protect your sensitive samples by building in mild and catch and release conditions, and how they can cut the number of steps in the process by being more selective and especially adaptable than standard approaches. And last but not least, how they can reduce overall costs with their very good stability through cleaning to ensure that you can use your columns multiple times. Let's start with the Clickmers. What are Clickmers? The term Clickmer is derived from click-aptamer. So those are chemically modified single-stranded DNA oligonucleotides. And they bind their target molecules adaptively based on their three-dimensional structure, which depends on their DNA sequence and the chemical modifications. So native DNA contains only 4 nucleobases, and that limits the mode of possible interactions, especially if you think of targets such as proteins, but also other biomolecules and small molecules. So that is where the modifications come into play. The initial idea was to use modifications that are designed to mimic amino acid side chains, frequently involved in protein-protein interactions. And we have now continuously broadened the portfolio of those modifications to also target other biomolecules or other molecular interactions. The point here is that the modifications broaden the chemical space of possible interactions. And so with these new interactions, we get binders with high selectivity and affinity. And in addition, we increased probability to develop excellent binders. We would like to take you through a couple of advantages using Clickmers for protein purification and in bioprocessing. So first of all, let's have a look at the molecules themselves. We mentioned the lower variation between batch material, and that is the result of the 100% synthetically manufacturing, which in addition offers high flexibility in conjugation. The manufacturing process is modular. In the first step, we introduce ethinyl desoxyuridine, EDU, into the DNA strand by a standard solid phase synthesis. And this is easy, scalable, GMP ready, and highly reproducible because there are no biological systems needed here. In the second step, the alkynes of the EDU are then used for acid alkyne cycloaddition, also called click chemistry. And while I would love to elaborate much more on this really cool click copper-dependent click reaction that you can see down there, let's stick to the most important facts here. We have a partnership for GMP manufacturing in place. It is cost and time efficient, and there are analytical methods for the QC fully established. Based on the synthetic manufacturing, there are different immobilization strategies available, which offers flexibility in conjugating the Clickmers to, for example, purification resins or nanoparticles or beads. The 5' and 3' termini of Clickmers can be labeled during synthesis with linkers of adaptable length and properties and functional groups, and there are diverse functional groups possible, such as amino group or thiol groups, fluorophores, biotin, and they can be coupled to differentially functionalized solid supports, having, for example, carboxyl groups or hydroxyl groups or streptavidin. On the right, there's an example of a coupling optimization, where you can see on the x-axis the input and on the y-axis the coupled Clickmer. You can see it's about 30% in this case, and that depends largely on different factors, such as protocol, support, or even the structure of the Clickmers. So after looking at these a little bit dry consideration about the molecule, we would like to show you how adaptable the Clickmer design process is to your needs and requirements. And for this, we have to take a look at the development process called ClickSelex. It is an in vitro evolutionary process of iterative cycles. What does it mean? You start with a very diverse single-stranded DNA library, so a lot of different sequences. Those are modified and will be incubated with your target. You wash away the non-binding sequences and recover the binding ones. They are amplified and modified and go into a next round of binding, washing, release. And throughout the selection, we systematically increase stringency, for example, by using competitors or increasing the number and duration of washing steps. And by this, we drive the Selex toward an enrichment of binders that are best suitable for the desired application. And the advantage here is that we can apply customized conditions, for example, during the incubation and recovery steps. So the Clickmers could be either tolerant towards changes in the surrounding microenvironment or bind under exact conditions, for example, a certain pH, salt concentration, temperature, or the presence of human samples like serum, blood, urine, or to release under specific conditions. So let's have a look at those two steps within the Selex that are customizable. For example, matrix requirements can easily be incorporated in the process. In this case, a complex matrix was added in the incubation step of the Selex to select for Clickmers that are that bind with high affinity in the presence of a complex matrix environment. On the right side, you can see an example data set. This assay is used throughout the talk to assess the binding of the Clickmers. It's an ELONA assay, so like an ELISA or immunoassay using the Clickmers as binders. And you can see in the control conditions a very good binding of the Clickmers to their target that is not influenced and robust against changes in the matrix, addition of serum or blood, for example. In order to select for high selectivity against a specific negative target, this negative target can be included into the process. Negative targets are targets that you want to avoid to be recognized, maybe because they are highly abundant in your sample or because they are very similar to your actual target. And here on the right side, you can see such an example. We are comparing here pools of sequences from the start, so here starting library and the round 10. And round 10 is expected to increase in signal if you have an enrichment. And that's exactly what you can see here for the target. And for the negative target, so for example, for this first one, you can see exactly the opposite, a de-enrichment, and particularly pronounced for the negative target number 2. So those two are two examples on how to customize the tolerance or include a certain specificity in the binding step of the Celix. Let's have a look at the release condition. In this case, the Click-Celix protocol was adapted to select for Clickmers that can be eluted with a high salt buffer, more specifically a high magnesium chloride buffer. So here the Celix was performed using this high salt or high magnesium chloride buffer to recover the sequences. And you can see here the same comparison as on the last slide. The starting library doesn't show a good signal. That's what it's expected because it's a lot of different sequences and it's not enriched. And when you look at the enriched pool, you can see a good interaction with the target. An incubation with the wash buffer here, control elution, does not decrease the signal. That indicates a slow off rate. And then if you add this high salt, it elutes the Clickmer almost quantitatively of the target. So the Celix process is adaptable for specific elution properties. In the beginning, we talked about how to protect your sensitive samples by building in mild catch and release condition. And you can see such an example here. Here on the left side, a target TBEV virus, NS1 protein, and on the right side, the dengue virus, NS1 protein. Again, in a direct ELONA format. You can see a binding from complex matrix in this case, and the binding is pretty resistant against deviations that you can see here in the gray boxes. In magnesium concentration, so going to higher or lower magnesium concentration doesn't change the signal much. And then the elution can take place either with low magnesium on the left side for the TBE virus NS1 protein, or with high magnesium here on the right side for the dengue virus NS1 protein. So that again shows really the selection adaptable to the needs. Let's have a look at the combination of binding and release conditions in this experiment. So here the Clickmers against the dengue virus NS1 were immobilized on a solid support. Then a target and complex matrix, in this case serum, was incubated with the Clickmer loaded beads. And the elution from the beads was performed using high magnesium. And on the right side, you can see in this silver staining that only the Clickmer and not the control sequence bind the target and releases it. The release is quantitatively, which you can see if you look at the two bands on the right side, the beads after elution that do not contain any protein. And I have to mention here that as a control, a scrambled sequence was used, having the same nucleotide and modification composition, but with different sequence. And that's a nice control that you can use throughout a development process. Yeah, with this, I would like to hand over to Steffi for the next advantages of Clickmers.
So thank you, Elisa, and we will go on with another important point, and that is the high stability of Clickmers. We have tested the Clickmers for heat and chemical stability. For the heat stability, we have cooked the Clickmers for 5 minutes at 95 degrees for 5 cycles, and we have tested them afterwards in an immunoassay. And as you can see in the graph on the left side, we can measure similar signals for each heat cycle compared to the nominal condition without a heat step, which demonstrates that we have no loss of performance after heating the Clickmers. The chemical stability might be of interest if you treat columns with sodium hydroxide between cycles. And here we have incubated Clickmers with varying modifications overnight in 0.5 and 1 molar sodium hydroxide. And the immunoassay on the right side shows that the measured ODs of each Clickmer are at the same level as the nominal condition without sodium hydroxide. And from these results, we can conclude that sodium hydroxide has no negative effect on the performance of Clickmers. For some applications, it may be necessary to use sterile material. So we have tested if the Clickmers survive autoclaving. And for this experiment, we have dried and autoclaved the Clickmers. And in the graph on the left side, you can see that the Clickmers react very differently. So there are Clickmers without performance loss, with a reduced performance, and Clickmers that completely lose their binding ability. And a reason for these differences could be the modification as well as the sequence of the Clickmer. And in this case, the stability should be tested for each Clickmer individually. We also want to show you the capacity of Clickmers to bind and dilute protein in a cycling binding assay. Here, the EGFR Clickmer has been immobilized on beads, and we have done 4 consecutive cycles of binding and elution. If you look at the silver staining gel, we can generate comparable protein bands after each cycle, which shows that the binding and elution capacity of the Clickmer remains stable over these cycles. So with this high stability through cleaning to ensure multiple uses of columns, the overall costs can be reduced as mentioned in the beginning. So another characteristic of Clickmers is the versatility in recognizing target structures. Clickmers can differentiate between highly homologous proteins. We have selected Clickmers for the NS1 proteins of the tick-borne encephalitis, the yellow fever, and the West Nile virus. And we have tested them in a direct ELONA assay on the 3 target proteins and on 3 additional NS1 proteins of the flavivirus family, the Japanese encephalitis, the Zika virus, and the dengue virus. And in the graph, you can observe that there are only signals for the NS1 proteins for which the Clickmers have been selected for, and no signals for any other NS1 protein. And that means that these Clickmers show no cross-reactivity towards any of the other NS1 proteins from the closely related flaviviruses, and that they are highly specific to their target protein. If your Selex target is a small molecule, then the Selex process is a little bit different from the standard Selexes, but can be easily adapted. And in this case, the Clickmer library is immobilized on a solid support via a capture oligo. The small molecule is then incubated freely in solution. And upon target binding, the Clickmers undergo a conformational change and are released from the solid support. And we have used this process to select for ofloxacin binders, and below you can see the affinity data of 2 ofloxacin binders, which show a good affinity in the micromolar range. Clickmers can also bind on targets on the surface of cells, and here we have flow cytometry data of a fluorescently labeled Clickmer that shows an expression level dependent binding. And these data show that we measure no signals with the Clickmer on target negative cells, but we observe a nice shift in fluorescence intensity on target positive cells in comparison to the control sequence. And here on the right might be interesting for some of you regarding safety concerns. We have tested this Clickmer for immunogenicity in PBMCs by measuring the levels of interferon alpha, CXCR10, and TNF alpha upon incubation with the Clickmer, and there was no response to the Clickmer observed. And in addition, below, you can see the tolerance in vivo, where mice were injected with escalating doses of Clickmer. A necropsy followed by organ inspection and weighing did not reveal any signs of toxicity. So finally, why choose Clickmers? We have learned that Clickmers are to 100% synthetic. They have a batch-to-batch reproducibility and low endotoxin levels. Clickmers can be designed to application requirements with a wide range of binding and dilution conditions in a fast development time. Clickmers are highly stable and have a versatility in recognizing target structures. So we are now at the end of our presentation. Thank you for your attention, and we would now like to start the Q&A session.